Sanasjatra and a good day. In this particular segment, we'll be talking about research and experimental designs. Experimental design is about how all variables and their factors are arranged and how they are supposed to interact in the study. We have been doing this subconsciously because the moment we arrange parameters to be analyzed in a statistical program, or maybe we line up for manual analysis, then we'll be constructing some degree of experimental design. The essence here is that the experimental design should comprise or serve three purposes. Number one is causation. Number two, the experimental design should have some degree of control over the outcome. And thirdly, it should show some degree of variability so that observation can be meaningful. Let's look at causation. Causation allows experimenter to make causal inferences about relationship between independent variables and dependent variable. Oftentimes, you can use incremental dosages or incremental treatment or intensity of treatment to see whether it results in the corresponding increase in response. Control will actually allow experimenter to rule out alternative explanations due to confounding effects of extraneous variables. For example, you may want to control for effects of the age by grouping young animals in young animals group versus old animals in old animals group. And of course, the third one will be variability, allowing us to visualize changes. They are meaningful so that we can relate the factors introduced to the outcome that they cause. Let's look at this particular example that I've been using again and again throughout this series of lecture. Suppose we postulated that extended periods spent at a mamak store will result in poorer semester results. In this case, you can see here, independent variables will be time at mamak store, whereas dependent variable will be semester results because our aim is to try to link the time spent at mamak store with the semester results. But as you can see here, the impact is actually not that straightforward. It could be confounded by other factors. For example, amount of sleep, the usual time of sleep, health status, and many other factors. So it becomes important for us to know how much of control that we have over our postulated study. You can design all the studies in the world, but if let's say you fail to control any of the variables, the study will become meaningless. So it becomes important if let's say in the following example, we can control all the factors and therefore we'll be able to get meaningful results as depicted in these three blocks here. In order to prove our hypothesis, then we need to have one group of people which spend long hours at mamak store, another group who spend shorter time at mamak store, and another group that never spend any time at all at mamak store. And all of them should be adjusted for sleep deprivation and making sure that everybody have the same amount of sleep pattern, and all other factors. So all the blocks in blue are actually the confounders that has been identified and controlled. Whereas the degree of time spent at the mamak store will be to some extent provide us with some degree of variability that we can observe by varying the intensity of treatment. And in essence, this will actually allow us to visualize whether time spent at mamak store will actually affect the semester results. As you can see, this design is not flawless. This design may have other confounders that you will identify over time. And this makes the defense of this kind of study sometimes very, very tedious and experiments very costly. So then we come to this particular consideration. The following should be addressed before considering any form of experimental design, because by considering all these factors, then you have a much better control over all the factors. So important attributes and features of experimental design should be, number one, there should be a presence of randomly selected or statistically equal control group. And then you should have a matched number of subjects so that you know that you are doing a balanced comparison. And where required, you may need to measure pre-exposure readings first, or the so-called baseline value. And then you may have multiple, one or more post-test assessment to see what will be the outcome of your treatment. And most important of all, you may need to introduce some degree of blinding into the trial. What is blinding? Blinding is actually a situation whereby subjects are not aware of their groups of researchers. Double blinding, on the other hand, is a special kind of blinding whereby subjects and researchers are not aware of the groupings. These are designed so that they will minimize biasness. Because being human, sometimes you may overcare or maybe overemphasize certain treatment groups, thereby massaging or subject certain treatment groups to unfair treatment, thereby skewing the results. Blinding is important because it is used to shield against Hawthorne and placebo effects. Hawthorne effect is actually a situation whereby the tendency of subjects to act differently when they know they are being studied. This is especially true if you are dealing with wild animal, because wild animals are not accustomed to human presence. So if, let's say, you are in the field studying the behavior, if you're not allowing time for them to adjust, you could be reporting the effects of your presence on their behavior. So this is actually something that we should be mindful. Placebo effect, on the other hand, is very important in the medical sector because it has been said that placebo effect accounts for about 30% of the drug effects observed in a lot of drug trials. Placebo effect is actually a tendency whereby some subjects tend to think that they are cured or sufficiently treated when they know about research being done on them. Well, in our day-to-day life, you may experience this. If let's suppose that you will have sickness and then you go and consult your friendly GP, and if your GP did not provide you a medicine, you may come back thinking that you may not have seen a GP and the pace of healing, pace of recovery may not be as fast when, let's say, that particular GP gives you some medicine to go home with. So this is actually an important effect of known as the placebo effect. Now, let's move on to specific parts on research and experimental design. We'll be covering around 6 designs, but I'll be emphasizing mostly on 2 or maybe even 3 designs that you'll be seeing every day. The first one will be completely randomized design. Second one will be randomized complete block design that you'll be using throughout your studies here in UPM. CRD is being used when you are investigating the effects of one factor and the data matrix arrangements follow that of one-way ANOVA. This is something that is important because you need to know what type of study you're using and then what type of data matrix that you need to know in order to key in your data. The second one will be randomized complete block design, which is actually a derivation of the factorial design. And this is used when you are investigating effects of one factor while ruling out the effects of the other factor. In this case, typically known as confounding factor. And this is actually represents a very important element in research design or element of control, as we already talked about earlier. Randomized complete block design can take into account 2 directions of change because it takes into account 2 factors, and the data matrix followed that of a two-way ANOVA in an SPSS spreadsheet. This is a simple CRD design. As you can see, you are comparing treatment effects across groups, and within groups, there of course will be experimental errors because animals being animals, they will not produce identical readings unless they are machines. This is actually the RCBD design, taking into account the effects of males and females. As you can see, the basic design is still more or less the same as the CRD, but with the added introduction of a gender factor here. So you can see there's one block for male and one block for females. And by introducing this, you technically have 2 factors in play, and you also double the number of animals, making the experiments much more time-consuming and costlier to perform. The third one is factorial design. Well, it can have 1 factor, 2 factors, 4 factors, and of course, 5 factors. SPSS only allows you to assess and analyze up to 5 factors. Anything more than that will be dimensionally very, very difficult for us to interpret. And in SPSS spreadsheet, it becomes important for us to understand that 1 factor is typically associated with 1 column in the data entry window. A very good example of a factorial design is actually the RCBD design that we just covered, whereby it takes into account a 2-factor design. The third one will be Latin square. Latin square is being used when you are investigating effects of two factors. They are minimally interacting with each other. It allows researchers to have more sample than usual, and however, it requires washout period. Well, the data matrix still follows a two-way ANOVA. Look at this particular example. The Latin square actually allows you to utilize a limited number of animals, something that is invaluable when you are dealing with limited number of samples. Let's say you're performing trial on the endangered wildlife, whereby you are only given very, very restricted amount of animals. So you can see in this case, this study only uses 5 animals. However, these animals are rotated across the 5 groups here. Okay, so by right, we should be able to rotate all these 5 animals, but it will require 5 rounds to do so. So in this case, you can see in round 1, animal number 1 is placed in group A, animal number 2 is actually in group B, and so on and so forth. By round 2, after a washout period, then animal number 5 will go to round A, group A, and so on and so forth. So you repeat this typically for 5 rounds until all animals are assigned to all groups. When you complete the 5 cycles that is being required, then you effectively will have 5 animals per group and 25 animals for the overall groups. So it becomes important for us to be able to utilize this. However, the shortcoming will be, this is actually time consuming. If let's say round 1 will take, let's say 1 month to complete, followed by a washout period whereby you ensure that all treatments are being washed out, that means without any trace of treatment, which can take maybe another month, maybe multiple months, then 5 rounds of treatment will actually set you back at least 10 months to 12 months. So this is actually not only costly, but it may not be feasible for certain other trials. That is actually time sensitive. Do bear in mind that by the time you repeated this particular trial, on the 5th round, the animal will have aged by at least multiple months or maybe 1 year old. So this is actually something that you may not be able to perform in certain group animals. For example, if you are examining the effects of nutrition on fast-growing animals, there's only a small window for you to perform this type of study, and therefore Latin square may not be the ideal design. The next one will be the split plot design, which is actually initially developed in 1925 by Sir R.A. Fisher, primarily for agricultural experiment. This style of design is dedicated to a situation whereby one factor can be easily changed while the other factor is difficult to change, as you can see in the subsequent diagram. So the hard to change factor will be sunlight intensity on the plots of land, because you can have plots of land that are open or maybe plots of land that are shaded by trees. So in order to make this standardized, of course, the ideal way is to chop down all trees and ensure all the plots of lands will have equal sunlight. Well, sometimes this may not be possible at all. However, it's easier to change the type of fertilizers used, brand X, brand H, and brand K, for example. So as you can see here, what you need to do now is just to assign the various plots of lands here as open, shaded, shaded, and open. And within each plot, okay, within each whole plot, you can then split the plot into 2, assigning half of it to be treated with fertilizer H or fertilizer K. And by randomizing it, you will get to a situation whereby all open or shaded areas are represented by both fertilizer H and K, allowing you to have a time efficient and cost efficient way of analyzing all factors that are in play in this experiment design. Of course, the variation of that split plot will involve the split block as well, which is depicted in a smaller degree of blocking of factors considered in split plot. The other way of dealing with research and experiment design will be, of course, the ever famous as the Solomon's design or the Solomon 4 group. This is actually a design that has been dedicated to single out subjects that has been sensitized to a treatment factor. For example, a person that has been exposed to a questionnaire before, or maybe a patient that has been exposed to anesthetic drugs or certain drugs, because repeated exposure to certain drugs may alter the liver physiology and therefore rendering the efficacy of the drugs to be different in animals in subsequent trials. So Solomon's 4 design will actually allow us to take into account subjects that have been sensitized to a treatment factor or a priming effect, as what they mentioned. So this will actually help to determine if repeated testing has an effect on a later test results. Well, that's why we're talking about questionnaires and repeated exposure to drugs. The only downside is that in order to do this, you need to incorporate 2 extra groups or maybe extra groups that denote pre-exposure and post-exposure, as you can see here. Whereas a typical experiment may only look at, let's say, group 3 and 4, whereby you have treatment or non-treatment. At Solomon's 4, as the name implies, will require you to introduce a pre-treatment group to be incorporated into the mix. So as you can see, if you only have 2 treatment groups, you need to introduce 2 more treatment groups that contain pre-treatment, making the overall total to be at least 4 groups. So you can imagine if you have 3 groups, 4 groups, so that will mean doubling, at least doubling of the Solomon's design. And in this case, since you are introducing a factor, a pre-treatment factor or a priming factor, the data matrix also follows that of a two-way ANOVA in SPSS.